Hi everyone, my name is Verusca Semmler Rossi and I am a master's degree student in the University of São Paulo, and I will present this work. It is in silico analysis of sugarcane SP80 3280 genes involved in metabolism of carbohydrates. Sugarcane is one of the most important activities of the Brazilian national agro-industry. It produces 80% of the world's sugar through the production of ethanol, and it is used in biomass to generate renewable energy. It has a very complex genome due to its polyploidy. With the advances in sequence technology, it was possible to produce two drafts for the commercial variety SP80 3280, but they are not complete but rather fragmented. These results have not been compared and integrated so far. Due to this, we identified the supposed complete and non-redundant set of transcripts and associated proteins or TAPs and carbohydrate active enzymes or enzymes in this variety genome, exploring the two genome sequences available for the same cultivar. To date, we have divided TAPs into families of transcriptional factors or TFs and other transcriptional regulators or OTAs. So what is TAPs and enzymes? TAPs are proteins responsible for gene transcription. They can be divided into TFs or OTAs. TFs are activated or suppress the transcription or target genes by the two systems. OTAs can be co-activators and co-repressors, which bind and influence TFs or recognize the main promoting elements or recruit components of the basal transcription machines, or chromatin remodeling factors. And enzymes are enzymes that synthesize or break down oligosaccharides and polysaccharides, and they have this sixth family. Here in this slide, we can see the pipeline we use to perform identification of TAPs and enzymes, where for sorghum, we identify these proteins without a redundant database, and why for the two versions of sugarcane, we perform the gene predictions without a redundant database and with a redundant database. And we also perform the genomic identification of all transcripts. And here, as we can see, there was the prediction of TAPs and enzymes following this process shown in the workflow. We identified 204 TF families represented by 16,057 genes and 71 OTA families represented by 4,245 genes. And for the enzymes, we identified 279 families. Here we can see the representation of transcriptome, and only 6.56% of the predicted transcripts were common to the two genome versions. And the figure C showed was the Busco analysis. Busco is used to perform completeness analysis, searching for a single copy preserved genes within the assembly. And here our results show to us that it's clear so that the gene representation for SP80 3280 is far to be completed. This is supported by the low redundancy seen in the TAPs and enzymes, and the whole genome and whole transcriptome when comparing both versions. For instance, we see in figure 4a that the genome from 2019 had 4 more sequence data than the version from 2017, only about 2 more predicted genes in the figure 4b, and a low fraction of identical predicted genes. Thus, this analysis highlights the importance of integrating the information from the two genome versions and thus have a better gene representation for SP8032A. Thank you, everyone.